So basically this is a forensic biology unit. Forensic biology is composed of two different sections, serology and DNA. Serology basically is where we identify what the body fluid is, whether it's blood, seminal fluid, hairs from a crime scene. Once we characterize what it is, whether it's blood or seminal fluid, it then goes on to DNA analysis. And in the DNA analysis, we'll actually extract the DNA out of it and eventually get to getting a DNA profile that will be compared against the question samples versus the known samples from both the victim and the suspect to say whether or not it's a potential match between those two. In here, we have 8 different work queues that are along the outside wall, and we also have 4 search rooms. And each one of the search rooms can actually be divided into 2 separate rooms, or the partitions can be opened up, swing to the outside, and be made into one large room. The advantage of this over our prior space is that it gives us 4 search rooms, 3 more than what we had previously, but it also allows us, once we open up the doors, we can move these 2 tables together and actually be able to look at a very, very large item. Beforehand, if we were looking at something like a California king-size sheet, that's a very large item, and to put it on one table, you'd actually have to fold it and kind of do sections at a time. This is actually our photo area. We had 1 search room at our old facility with 1 table, and what they basically have to do is get a stool or some sort of a ladder and actually stand on top of that using a regular camera to try and take a photo of it. What's nice about this system, though, is that we can actually, as I stated before, we can put a large item down on these tables, and the camera will actually run through the computer that you're standing in front of, and it'll give us a live view of the image. So we can do all focusing as far as moving the camera around. And these are actually on rollers, so the entire system rolls from side to side, and then the cameras will roll forward and back. And then all the lights are the same way. They're on individual rollers, so we can adjust the lighting, adjust the camera exactly like we need it. Then we can take the picture of it. It stores the image in the computer. We can put it out to our network, go back out to our work queue and pull that image up and import it into our case notes if we want to. So these are the Nikon CoolScopes, and basically what the CoolScope is a microscope in a box. So what Nikon has done is they've taken all of the objectives and everything from traditional microscopes, put them inside of this box, and they've taken the actual eyepieces off. So what it does is it runs through a computer or uses the built-in firmware and uses a monitor. So what we do is we actually take the slide, put the slide into the CoolScope. It projects it up onto the monitor for us, so it reduces eye strain on the analyst because a lot of time is spent at the microscopes looking for the spermatozoa on the slides. So it reduces the eye strain, reduces the time because it's basically a point and click as opposed to actually scanning the entire slide. The other advantage of this is we can take immediate pictures of it. It's got built-in, we can use compact flash cards, or we can tie it into our network, which is what we're trying to do. This is a 7500 real-time instrument. This instrument will ultimately give us a report that shows us specifically how much DNA is in that sample. We have to know how much, or else we overload our system, our instruments won't work properly. So once we've quantitated the amount of DNA that we have using the 7500 instruments, the next step is to come in this room. In our old laboratory, we actually had one of these hoods, so it was shared amongst all the DNA analysts. With this room, we developed it with 5 hoods so that multiple people can come in here and set up their samples at the same time. This is essentially a clean room for us. Once the DNA comes in here, we mix it with the reagents that are actually going to allow the PCR process to happen. Once they're mixed with those reagents, then they're put into the pass-throughs, which you see on the wall to the left here, to my left. Now what happens is we put them in the pass-throughs, and then the analyst can leave this room and go into the next room and actually retrieve it from those pass-throughs. And then once they've ran those samples, all of the material that's used in here, such as the trays, can be put back through the other pass-through and actually UV. And the UV actually breaks down any DNA that's present in the samples. So once we set up the samples in the other room and send them through the pass-through, they come to this final room. And it's called post-amp or our instrument room. Basically what happens is we'll bring the samples in, and they'll go onto the small instrument that's there to your left. It goes through different temperature ranges, takes those samples through those different temperature ranges, making copies of that DNA. So where you might start with 1 strand, it's an exponential growth over 28 cycles. You may end up with 1 million or 1 billion strands, copies of that DNA. Once we've amplified the sample, then we pull it off, we set the sample up and get it ready to go onto the instruments. We actually have a gowning and de-gowning vestibule just for this room. And the reason we do that is because once the samples come in and are put onto the 9700 thermocycler and that sample is amplified, at that point you have amplified DNA. But that DNA has the potential to contaminate other DNA very easily, because amplified DNA can be amplified again easier than regular DNA. Something else that we put into this room are the Niederman hoods that you actually see above the instruments. The advantage of those instruments actually give off a lot of heat because they have a built-in laser inside of them. They also have a camera. Both of those give off an extraordinary amount of heat. In our old laboratory, what happened is all that heat was just filtered into the room. So we had just extreme temperature changes in that room from when an instrument would kick on to when it would kick off. So what ends up happening is these instruments actually compare those samples to what's called a size ladder to actually give us a profile. And we usually run a ladder about every 10 samples. Well, in the old lab, with the temperature changes, sometimes you'd have to run that ladder every 2 or 3 samples just to be able to get it close enough to the sample so that they ran about the same amount of time. What we've noticed in this room since we've been here, the 3130s actually keep track of the ambient temperature in the room. So you can actually pull up a screen and it'll show you what the temperature was in the room through the entire run. Where at the old lab, we were seeing a fluctuation of up to 7 degrees in 1 minute, which is a huge fluctuation for us. With these, since we've been in here, they fluctuated less than a half a degree over the entire 30-minute run. So, I mean, as you can see, the hoods along with the new heating and air system, everything has made this room a lot better for the analysis.